Thawing is the process of allowing a frozen substance to return to its natural state. In context of meat, thawing involves reversing the freezing process to make the item ready for consumption. Meat would undergo quality deterioration such as oxidations of lipid and protein denaturation, moisture loss, color deterioration, flavor loss, textural changes, as well as microbial spoilage during freezing and thawing process. A suitable thawing method can greatly reduce the loss. There are some quality issues during thawing of chicken breast that can arise due to various factors, which is freezer burn. The problem that occurs is chicken is not properly sealed before freezing. The solution for this problem is use airtight packaging or vacuum-sealed bags before freezing and thaw using recommended methods. Next, the quality issues when thawing chicken at room temperature for an extended period may promote bacterial growth, and the solution to this problem is thaw chicken in the refrigerator or using other safe and rapid methods. Lastly, improper thawing methods, such as using a microwave with high power, can lead to changes in the texture of chicken breast. The lower microwave power setting have to be used to overcome this problem for gentler, the thawing method like refrigerator to maintain the desired texture. The objectives of this experiment were to evaluate the effects of thawing process on frozen chicken carcass and to measure the quality loss after subjecting with different thawing methods. The experiment was started with cutting the fresh chicken breast into the same size and weight all the samples. The samples were packed into polyethylene bags and labeled the control and 3 experiment samples. All the 4 samples were sealed properly using impulse sealer. The samples were stored in the freezer overnight. There were 3 treatments for thawing, which are treatment 1, microwave thawing, treatment 2, tap water thawing until meat totally defrost, and treatment 3, low temperature thawing, chill at 4 degrees Celsius overnight. After thawing, the samples were analyzed, thawed to determine the quality loss. First analysis was determination of color for every treatments. The colorimeter was calibrated and the lightness, redness, and yellowness was recorded. After that, another analysis was determination of pH. The minced breast chicken were taken and stirred with distilled water. The pH was measured using pH meter. Lastly, final analysis was water holding capacity. Chicken breast was placed in between Whatman paper then. It was covered with aluminum foil. 2.5 kilon of load was pressed on it for 2 minutes. The result obtained as the table below. The study measured the surface color of chicken meat using a chromometer, revealing that metmyoglobin accumulation during storage significantly affects its discoloration. Meat. The water holding capacity of meat is determined by the structure of myofibrillar proteins, specifically myosin. Freezing and thawing can alter water content and distribution in meat tissues by releasing water immobilized and linked to protein in intracellular spaces. Among 3 kinds of thawing methods, treatment 2 had the lowest thawing loss, 1.59%, and treatment 3 had the highest thawing loss, 5.04%. The differences in the thawing loss for various methods are due to the freezing and thawing process, which alter the content and distribution of moisture in muscle tissue. The thawed meat samples had a lower pH than the fresh meat, due to changes of the ionic balance within the meat. When microwave thawing meat and poultry, it is recommended to plan to fully cook it immediately, because some parts of the food may become warm and begin to cook. Holding partially cooked food is not recommended because any bacteria present would not have been destroyed and may have reached optimal temperatures for bacteria to grow. In a study, all the evaluated thawing methods affected meat characteristics and chicken breast thawed under refrigeration, compared with thawing in a microwave oven and at room temperature presented the best meat quality results. However, In this present study, the results of refrigeration sample not closest to fresh samples, which might be due to exposure during thawing process, can contribute to oxidative changes in the chicken. This may affect the quality loss of chicken.